Oh, EGPET1 Tetrahymena gene expression in response to diet. This research is done by Nana, Lauren, Emma, and Ben.
Our research question is, what happens to the expression of lipid droplet protein genes in fat-fed cells? We are specifically looking at the relative change in gene expression of the AGPAT1 gene when the cell is in a fat-fed diet, oleate, versus when the cell is given a normal lab diet, NEF. In Tetrahymena, the AGPAT1 gene encodes for a EF hand protein, and this EF hand protein is really important for the uptake of calcium in the cell. In mammalian cells, the AGPAT1 gene is involved in the de novo synthesis of triacylglyceride, also known as TAG. AGPAT1 encodes for an enzyme that converts lysophosphodietic acid to phosphodietic acid. PA is known to be a common intermediate for the synthesis of TAG. TAG is involved in the storage and transport of fuel and lipid proteins. TAG storage within tissues takes place in the form of lipid droplets, and lipid droplets are linked to many cellular functions, such as acting as a reservoir for transcription factors, histone proteins, and acting as a platform for viral replication and for protein degradation.
So first we had to test that our primers for the AGPAT gene worked. So after doing a PCR with the specific primers, we then loaded that genomic DNA into the agarose gel and conducted an electrophoresis. The picture on the right shows the AGPAT on the leftmost side of the gel next to the loader DNA, and the picture on the left is a reference for how it should look like. The reference shows that it shows that AGPAT was expected to travel to around 250 base pairs, and our products also traveled all the way down the column. They went slightly farther than 250 base pairs, but were still in the general area. And overall, we got the expected gene amplification, and it showed that the primers worked.
So before going into the analysis of our own CT data, I just wanted to give a quick RT-qPCR overview as a quick reminder. So this is a representative cycling graph given to us by our lab instructor, and it's just data pulled from across the labs in this experiment. And so the CT value, or the cycle threshold, is essentially the number of cycles required for the fluorescent signal to cross the threshold level, which essentially means it's exceeding the background level. So you can see where the lines cross that line, that would be their CT value. And the CT levels are inversely proportional to the amount of target nucleic acid in the sample, which in this case, we're concerned with the AGPAT1 nucleic acid material. And so now for the analysis of our own data, this is a graph that we made using the raw CT averages from our lab. And it's comparing the 3 different genes in Tetrahymena related to lipid droplets and their expression relative to being fed oleate or the NEF. So we're focusing again on the AGPAT1 gene, which you can see is represented by the middle bar. And so the oleate fed had a very, very slightly higher CT average than the NEF fed Tetrahymena. And this is represented by the fold change. And the fold change is essentially like the ratio of change between the oleate fed CT and the NEF fed CT values. And so when the fold change is above 1, that would indicate an increase of gene expression. And when it's below 1, it would indicate a decrease in gene expression. And so our fold change value was calculated at 0.96, which indicates just a very, very slight difference or decrease in gene expression in the oleate fed versus NEF fed. However, given that the value is approximately 1, the decrease is pretty much statistically insignificant and doesn't really represent little to no change in the gene expression.
We also looked at the MELF curve. So that was looking at using cybergreen as our fluorescent indicator, which the relative fluorescence can be seen on that y-axis, whereas the temperature at which the denaturing occurs can be seen on the x. So our PCR genomic template was the cDNA. So we conducted a reverse transcriptase quantitative PCR, and we found that given our peak of the AGPATs was within the same temperature range, So approximately 76 to 80 degrees is when the double-stranded DNA fell apart, that there was little contamination evident except with the one exception of the peach-colored egg PET sample, but otherwise we were relatively clean. And then it was also very easy to have gene differentiation, and that each of the three obvious respective humps were correlated to their own genes. So egg PET was in the middle with that 78, 76 to 80 degree range. So our conclusion, an oleate-rich diet bears little to no impact on the expression of the egg PET gene 1 expression in Tetrahymena thermophila when compared to gene expression with a regular NEF diet. So our hypothesis is that oleate feeding does not affect gene expression of egg PET1 because egg PET genes encode necessary proteins for cellular metabolic stability, and thus the gene needs to be constitutively expressed for the stability of the cellular functions, regardless of the available diet.
Our evidence for this is that the AGPAT1 gene in mammalian cells encodes for a really important enzyme that breaks down, that creates a really important acid, which is then super important in the synthesis of triacylglyceride. And that tag affects the overall functionality of the cell, and the proper distribution of the phosphodietic acid is necessary for the synthesis of that tag. Because tag is so important and it's involved in the storage and transport of fuel in the form of lipid droplets, and those lipid droplets are super important because they are linked to really crucial cellular functions, as mentioned earlier, as acting as a reservoir for transcription factors, histone proteins, acting as a platform for viral replication and protein degradation. So this is why the AGPAT1 gene would be constitutively expressed. Another reason that makes us so that supports our hypothesis is that in Tetrahymena cells, the AGPAT1 gene is also known to encode for a protein that's really important for the uptake of calcium. Without that uptake of calcium, it would be pretty problematic. So this is another reason why the AGPAT1 gene would be expressed constitutively.
And these were our sources. Thank you for listening.